can move for the next speaker. We have Dr. Allyson Rosen from Stanford. Allyson, are you with us?
Yes, and you're going to have your fastest finger because I have lots of slides which are largely animation. But I'm going to talk about TMS implementation, specifically understanding clinical targeting and precision, and I couldn't have had a better introduction from Mike Fox. It's been an inspiration. Next slide. So these statements are my own, and yes, so this paper provides a lot of detail about the analysis we conducted, and so I refer you to that by Valerie Cardenas. Go on to the next slide. And I want to talk about this study that's been very misunderstood. It's Savage's RTMS for depression in veterans. It was a $12 million clinical trial, sham-controlled, next slide, that showed no active sham difference. And it's a cautionary tale why precision matters. So I, back in 2012, started an imaging study in this exact population, looking at targeting precision. Next slide. So we all know that clinicians have this problem where we have this relatively focal treatment, and yet we can't see inside the skull. Next slide. To do that, we need image guidance, and that isn't clinically available. Next slide. But there's this vast number of people receiving therapy clinically. They're trying to target the DLPFC, classically identified as 9 and 46. Next slide. And in this study, we used the 6 centimeter rule, identifying the motor hotspot and moving 6 centimeters forward with the hope to avoid the premotor cortex. Next slide, which was basically our understanding of why the therapy didn't seem to work. They used this template, next slide, and traced out on the skull the 6 centimeter rule. Next slide. But as you know, head size variability matters. So 6 centimeters on Mount Rushmore is different from 6 centimeters on a quarter. Next slide. We used an approach pioneered by Tal Herbstman, in which we placed markers on the motor hotspot and the clinical targeting point. Next slide. And just like Tal Herbstman, we found variability in the regions targeted, and these are the templates of the DLPFC that he used. Next slide. We published this in a separate paper. But I knew back in 2012 when my grant was funded that we needed functional MRI because there was far more to this. Next slide. And we used functional connectivity. You all know what networks are. We did see voxel correlations with the clinical targeting region, which I'll present next. And so that our patients' networks are similar to the canonical networks. That was, one reviewer was concerned that depressed patients didn't have the same networks, so we got the same ones. This is a triple network model networks, but we have more in the paper. Next slide. And again, you'll notice that the more posterior targeted patients tended to be more likely to be, quote, non-responders or have a different response, similar to what Herbstman showed, that more anterior and lateral seem to have better responses. Next slide. But we compared it to more modern atlases, for example, the Human Connectome Project by Glasser and the Network Atlas of Yo. Next slide. And so on the left, you see a analysis similar to Tal Herbstman's. On the middle one, you can see what we presented relative to the Human Connectome Project. And we also show the symbols, in the symbols, the sham and the active average regions. And so you can see that the non-responders tended to segregate out into an area that was based on the Human Connectome Project as area 8AV. And the other conditions tended to be in the classical DLPFC, according to a Human Connectome map regions. And then if you compare that to the Yo Atlas, the non-responders tended to be stimulated in the default mode network. You can see that in purple here, whereas the responders, the active responders, tended to be in more classical areas. And you saw this difference only in the active condition and not the sham. So that further supports that location matters. Next slide. But if you take this average responder versus non-responder, and then you do a seed voxel analysis, go on to the next, and compare the derived maps in all the patients, you find that the non-responder network derived tends to be more in the default mode network, and the responder tends to be more in the salience network. And again, this was in a separate paper. Go to the next slide. And it just aligned beautifully with Shan Siddiqui's axiosomatic versus dysphoric target maps, so that the non-responders tended to be in the axiosomatic region, and that aligned nicely with area 8AV. Next slide. So in order to move beyond that and to look at the networks targeted, often clinicians think about what distance rule to use. We identified the motor hotspot using the usury hand knob and then moved forward centimeter by centimeter. Go next. Specifically, we did a seed voxel at each clinical fixed distance rule ROI. Then we averaged the derived connectivity maps across patients, and then we are generating separate maps for each rule. In this next series of slides, you can just cycle through. And really the point is that the networks, so you can see in green the variability in the regions across patients. This is the 5, go to the next 6, 8, 9, 10. And so you've gone the expanse of that classical DLPFC, that the networks vary likely. Next slide. In that paper, we also traced out the BMF3, and you can see next slide, the network there. So we all know, based on TMS fMRI studies, that not only you target likely different networks. Next slide. And if you look at the dispersion among patients in the regions directly targeted, the fixed distance rule have a lot more variability across patients. The applied 6 centimeter with the fiducial markers had even more, likely due to operator variability, whereas the head size adjusted rules did much better, less dispersion. Next slide. And we looked at this dispersion and the variance accounted for. And so a large part of that is from variability in identifying the motor hotspot, but it's not completely accounted for. There's probably some operator variability as well, and we've known that for a while out of Alvaro's work. Next slide. So just looking at the areas directly targeted by these fixed distance and adjusted rules, you can see going from left to right in the fixed distance rule, if you're at the 5 centimeter rule in orange, you can see you're targeting transitional DLPFC. And then as you move forward, you're more likely to be in the classical DLPFC. And again, these are HCP regions. Whereas in the head size adjusted rules, you tend to be in the classical DLPFC. A very small number of patients are targeted in the premotor region. Next slide. And if you look at the networks targeted on the 5 centimeter rule, you tend to be more in the default mode region. As you move farther forward, that shifts more to the salience network at 8 centimeter, and then more to the frontoparietal network. Interestingly, the BMF3 has half frontoparietal and half salience. Next slide. We also looked at connectivity with the subgenual ACC, next slide, and we included your coordinate from 2012, Mike. So the 5 centimeter rule did not have very strong connectivity with the subgenual. Next slide. Now we're doing TMS fMRI, looking at that area 8AV. Next slide. So in summary, locations targeted matter likely. Posterior targets more disproportionately target the default mode network and transitional DLPFC. More anterior targets the salience and then frontoparietal networks. Classical DLPFC are more dominant, and likely head size differences, operator error, motor cortex variability, all contribute to variation in the regions targeted. So next slide. And thanks to all my collaborators. So hopefully I've sorted out the misunderstandings about that VA clinical trial.